In a previous CRISPR video, I told you that to save Cage's life, scientists edited his genes themselves. But if we could make our own DNA, would we need any editing? Think about it. If one day scientists sit down and start literally making human DNA on a table, not inherited from parents, not extracted from any cell, but made from scratch, molecule by molecule. Until now, you have heard about tools like CRISPR or gene editing. But what's happening now isn't editing. This is writing the human genome from scratch. And this story is starting from Cambridge's Molecular Biology Institute, where scientists have recently claimed that they can make human DNA themselves. Not the whole thing yet, it's just the beginning. But this beginning could change the future. By the way, if you haven't watched my CRISPR video yet, then first go watch it from the link given in the description or via the I button on the top, so you'll understand this video even better. So in 2003, when the Human Genome Project was completed, there was a stir all over the world because for the first time, scientists decoded the human genetic code, meaning the four bases in all our DNA: adenine, thymine, guanine, cytosine. A for adenine, T for thymine, G for guanine, and C for cytosine. Their complete blueprint was revealed. This was biology's kind of moon landing moment because it was a very big discovery in the medical field. But the matter was limited only to reading the genome. Now what Cambridge's team is doing is the next step: writing DNA, and that too from scratch, meaning completely from zero. Now the question arises, what does synthetic genome mean? You already know what synthetic means, not fake but lab-made. Just like we write new software, scientists are now coding DNA in the same way. In this project, they first made DNA only for small organisms, like bacteria or yeast. But now, the plan is to make small parts of human DNA, meaning chromosomes. This has been named SynHG, meaning Synthetic Human Genome. Under this project, scientists will connect the human genome with chemicals, manually assemble base pairs to create DNA strands. According to reports from BBC and The Guardian, Cambridge's MRC Laboratory of Molecular Biology has officially announced that they are going to synthesize the human genome. This doesn't mean that a synthetic human will be made tomorrow. No, right now it's starting with basic chromosomes. The entire research is being supported by the Wellcome Trust, which has recently given a grant of £10 million. The project leader is Professor Jason Chin, whose team has previously written the genome of E. coli bacteria. They have designed bacteria that can resist viruses. Now imagine applying the same logic to human cells. Now, many people might have this question in mind: is this like CRISPR? So the answer is no. This is next level from CRISPR. CRISPR is an editing tool, like correcting a spelling mistake in a book. But synthetic genome means writing the entire book yourself. However, both technologies might merge in the future. Like with CRISPR, you'll modify the gene, but with synthetic biology, you'll decide what should be in the gene. This is a very big difference. Until now in science, we have made many synthetic organisms. Scientists have synthesized yeast chromosomes. E. coli's genome has been rewritten. Synthetic cells have been made that live only in the lab. But when it comes to humans, until now, only small genetic circuits and mini chromosomes have been made. SynAG's aim is that by joining small parts together, one day we can make the entire human genome ourselves. Now you might be thinking, are we trying to become God? Won't there be any misuse? But the most important thing, what will be the benefit from this? So in the next part of the video, we'll talk about this research's possible future, ethical concerns, and real-world applications. What uses it can have in the future, its ethical questions, could this be a threat to humans? And finally, our opinion plus simplified conclusion. Now just think, if we can design human DNA ourselves, what would that mean? Many genetic diseases are inherited from parents, like thalassemia, Huntington's disease, or muscular dystrophy. If we design DNA from scratch, then we can remove such diseases from the start itself, making virus-resistant cells. Just like Jason Gene's team made bacteria virus-proof. In the future, we can design human cells in the same way that recognize virus like HIV or flu and neutralize them themselves. Future of organ transplants. With the help of synthetic genome, we can make such organs that don't get rejected and can also fight diseases. In the future, heart, liver, kidney can be made in the lab, fixing mitochondrial disease. DNA isn't only in the nucleus, but also in mitochondria. With the help of synthetic biology, we can make new mitochondria that can stop inherited metabolic diseases. But just like every technology has two sides, this one does too. Many experts fear that synthetic DNA could be misused. In the future, biohackers could make designer babies whose intelligence, looks, height are decided in advance. Will rich people be able to design their children's genes and poor children remain normal? This sounds straight out of sci-fi, but scientists and ethicists are already discussing it. One more big fear is of bioweapons. If any group can make a synthetic genome, then they could make a virus in the lab that targets a specific population. For now, this seems extreme, but the history of biological warfare teaches us that misuse of technology is possible. That's why, along with this SynAG project, Cambridge and other organizations have created ethics panels that will keep a check on every step so that nothing becomes uncontrolled. Now, a philosophical question. Should we even do this? If humans can write their own DNA, then is it taking the place of God? Some people consider this against nature's laws, but others say if lives can be saved through science, then it should be used. That's why this project includes not only scientists, but also ethicists, philosophers, and legal experts. This isn't just a question of biology. It has become a question of society, politics, and ethics. Until now, we have been reading DNA. We learned to edit DNA with CRISPR, but now, with synthetic biology, we can design DNA ourselves. This isn't just a scientific leap, but a civilizational leap. If done in the right way, then this technology can end cancer, genetic diseases, and virus outbreaks. But if misused, then it could become the most dangerous tool in human history. Now the decision is in our hands. Will we use it for the good of humanity or leave it as a threat to humanity itself? Tell in the comments, do you support the future of synthetic genome or oppose it? Is it necessary or risky? If you found this video valuable, then like it, share it, and don't forget to subscribe to the channel for such deep scientific based videos.